My name is Xiading, and from Nanchang University of China. Today, I'm happy to present our project, the impact of aquaculture system on the microbiome and gut metabolism of Chinese softshell turtle. The work was published in the Journal of iMeta. We know that microbiome is important for the host fitness. Farming practice may reshape the host association microbiome. Rice fish culture is a good way to produce grain and animal protein in the same place. We want to know how the agricultural practice impact the animal microbiome, which may markedly affect the successful farming of the host. Here, two different agricultural systems, rice fish culture and intensive pond culture, was compared to analyze the skin, oral, and gut microbiome, the metabolism, as well as phenotype of the Chinese soft-shell turtle. First, we observe the microbiome structure. Higher alpha diversity was found in RFC than those in IPC. At the same time, the functional redundancy was also greater in RFC than those in IPC. And the agricultural system have the strongest influence on the gut microbiome, followed by skin microbiome, and finally the oral microbiome. Next, we make a source tracking analysis. The microbial community in RFC originated from more unknown sources than that of IPC across all body regions. This may partly explain why the microbial community has a higher alpha diversity and functional redundancy under the RFC. Then we analyze the microbial community assembling process. We found that the oral and skin microbiome of the RFC exhibited a significant higher proportion of generalists and broader habitat microbes than those of IPC, but not the gut. The oral and skin microbial community assembling of the RFC was governed by a significant greater proportion of deterministic process than that of IPC. However, the gut microbial community assembling was governed by stochastic processes in both RFC and IPC. It meant that microbial community assembling in different agricultural systems was tissue dependent. Next, we observed the phenotype. You can clearly see that the body size of the Chinese soft-shell turtle under the RFC is observably smaller. In particular, we found that under RFC, the liver significantly accumulated more lipid droplets. Then we focus on the gut. It's interesting. The microbiome that fermented polysaccharides was enriched in the gut in RFC. So we further examined gut metabolites. There was a higher concentration of fatty acid, TCA acid, and monosaccharides in the gut of RFC. Finally, we identified the king gene and the microbe contribution to find significant change in the gut metabolites. Together, different agricultural systems induce different tissue-specific microbiome assembly that change host fitness in part through gut metabolites. Thank you very much.